there. It looks like it's about 11 o'clock, so we're going to get started. Thank you all for joining us today for our webinar. My name is Jackie Carville, and I'll be coordinating the webinar today. I'm here with Matthew Kaiser, who will be going over sequence editing and annotation within DNASTAR's Lasergene Genomics Suite. You may have noticed that your phone has been muted. However, we do encourage you to ask questions along the way. To ask a question, just type it into the chat dialog and select Send to Host. I will then direct these questions to Matt to be answered for the whole group. If you need any assistance or have any questions during the webinar, you can send a chat message to me, email me at webinars@dnastar.com, or tweet us at the Twitter handle @DNAStarInc. And with that, I'll go ahead and turn it over to Matt.
Thank you, Jackie. I'll switch over to my desktop here. Just a moment. Okay. Well, welcome everybody, and thanks for the intro, Jackie. Today, I'd like to do a webinar that discusses some of the more advanced tools that we have in our SeqMan Pro software that involve editing at different levels and also some different annotation options. So I've been working with customers here for almost 10 years now, so I have a pretty good idea of some of the features that I think are really useful, and in some cases customers might miss or might not be aware that they occur in our software. So hopefully we can cover some of those topics, and of course I'll take questions at the end, so if there are things that I missed or didn't explain clearly enough, you'll have a chance to get your questions answered. So before we start, as we like to do, we'd like to give a little background on DNASTAR as a company. I know there are some of you that are familiar with our company and some of you are new, and this might be your first webinar. We are located in Madison, Wisconsin. I can assure you the weather looks absolutely nothing like this today. It is snowing out, and there's over 2 feet of ice on Lake Monona. Hopefully in about a month and a half, though, it'll look like this. So fingers are crossed on that front. So our company was founded by Dr. Fred Blattner. Fred and his lab were genomics pioneers and sequenced the E. coli genome back in the 1990s. So the software that they developed in the lab was commercialized, and ever since it's really been a pioneer and used by our customers for large projects like genome assemblies, and as well as smaller projects like assembling things like PCR amplicons. SeqMan Pro in particular was a very important tool for putting together these genomic sequences. And so SeqMan Pro has a lot of different options. For those of you that have worked with SeqMan Pro and looked at some of the menu options, there's many different things, many different ways to edit sequences, different ways to assemble sequences, and it can be a little bit overwhelming. And so the hope is today we'll be able to go through, and I'll try to outline the functionalities of SeqMan Pro, and we can kind of go through some different workflows. And so this software then was really designed out of a necessity for genome sequencing. And when it was commercialized, it was we were able to have it run on desktop computers, now more recently up on the cloud, and hopefully to make these very powerful tools accessible to all of you. Our software is research grade. It is the most cited bioinformatics software in literature, and so you can see right now we're a little bit over 9,000 citations, which we hope that trend continues. And so the support that we provide with the software is wide-ranging. I spend most of my time working with customers, both customers who have purchased our software and have questions, and as well as customers who are evaluating software. My focus has been next-gen sequencing technologies. And so some of the data that we'll look at today will be NGS, but we'll also spend some time with some Sanger data. The support also includes things like our website, which has numerous training videos. So there's, and this is just a screenshot to show all different types of workflows, and they're short videos between 1 minute and 5 minutes long on all different topics. And you can see under next-gen workflows, there's de novo assembly and ref-guided genome assembly. And so we'll be talking about those workflows a bit today and looking at some of the tools. There are also, of course, monthly webinars like the one that we're doing right now. So we try to do them once a month on all different topics. And these are different people here. It looks like Jackie did the last 3. So December, January, February on 3 different topics. And so if you have additional workflows that you'd like to learn more about, you can certainly watch these up on YouTube and learn more about our software. So what we're going to be talking about today is SeqMan, and that both SeqMan Pro is where all the editing functions will be, but we'll also look a little bit at SeqMan Engine. And so these are softwares that are part of the Lasergene Genomics Suite. And so the suite will include the assembly and the downstream editing and analysis tools. Of course, DNASTAR offers other packages that include things like phylogenetic alignments and structural biology and protein folding. So again, depending on your needs, you may be interested in a sub-package or the entire DNASTAR suite that includes all these sub-packages. So kind of the topic for today is really SeqMan Pro and the functionality that it provides, and I'm going to try to explain the 2 different modes that SeqMan Pro is used in. And this is it can be a bit confusing because there is one interface that does 2 quite different things. And the 2 modes are the editable SeqMan files, the SQD files, and then the other mode is the uneditable BAM.assembly files. And depending on the type of project that you have, you may have to use one workflow or the other, or you may have the option to produce both types of files from, say, a SeqMan Engine assembly. And to kind of describe it in a little bit more detail, the editable SeqMan.SQD files, when we're in this mode, this allows the user to do things like assemble Sanger data. So this is the you can drop Sanger data right into SeqMan Pro and align the data. You can trim the Sanger files. You can view the chromatograms, do micro-editing, generate a consensus, edit the consensus, export the consensus from SeqMan. So a common workflow would be a de novo genome assembly of a bacteria. You want that to be editable, so you can edit both the sequence data that's aligned as well as the contigs that are formed. So then there's a little higher level editing. So rather than single sequences, you can assemble and edit contigs, which are clusters of sequences. So I can break them apart. I can edit the ends of the contigs and improve them from the assembly. And then there's another level of editing, and that's the scaffolding. So I can take contigs and I can order them into scaffolds, and I can also have the ability to edit the scaffolds. So there's different levels of editing that are possible in this SeqMan file. That allows us to do things like genome closing. So there's additional alignment algorithms that allow us then to close gaps, also to add annotations, and we'll look at that as well. So a part of the workflow for example, for a de novo genome assembly is to do all the editing, create scaffolds, close gaps, add annotations. Now there are limitations to this workflow in our software. The limit now is about 15 million reads. And so that covers bacteria, the smallest eukaryotes. But if you have larger projects, small to mid-sized eukaryotes, you wouldn't be able to necessarily load the whole genome in at one time. I have customers that might work on one chromosome at a time that fits in this size. So it's just something to be aware of, is that there is a size limit here, and you also get some performance issues that we can talk about, that as these files get close to the limit, there are certain algorithms and certain ways to edit that fall more in line with how the software functions than some of the others. Now, the other mode is the BAM mode, and we've got lots of webinars on our recorded on our website that focus on this new BAM file. And the idea with BAM files are that SeqMan becomes really more of a viewer, a way to look at assembled data rather than an editor. And so a common example would be if I have an RNA-seq or a genome alignment to a human genome that I've done in our SeqMan Engine software, that might produce a file that has chromosomes that are over 100 megabases long and may contain hundreds of millions, even billions of sequence reads. So they're gigantic files. It's not possible to edit those. All these edit points that you have in a fully editable SeqMan file, add quite a bit of overhead to file sizes and efficiency. So BAM files are great in that There's no limit to the size really, but they're not editable. And this, so SNP and variant analysis is usually done in this kind of a workflow, RNA-seq. And again, the capacity, we've had data sets with close to 10 billion reads in an assembly. So we'll think of this in two different modes, and I'll show this a little bit more in SeqMan Engine. We're going to focus most of today on the editable version of the SeqMan file. And so the editing tools, again, and I'll try to go through these in the software, micro-edits, which are individual bases within individual aligned sequences, or micro-editing in the consensus, again, making human corrections on bases, and then more wide-scale editing, contigs and scaffolds, and then merging contigs together. And there's lots of different options within these workflows. Then we'll move on to some of the annotation options. And so annotations can be created in SeqMan Pro 4 different ways, maybe even 5, but I think we'll talk about 4. One is manually creating features, so we can add a feature manually. Another way is to transfer features from a reference sequence to a consensus. So a common example would be I use a reference genome to guide the assembly of my data. The reference genome is annotated. In mass, I can transfer those annotations over to the consensus for my new organism. I can also, SeqMan will also auto-create features based on coverage information. So different metrics from the coverage table, for example, regions that have high coverage or low coverage, or regions where there's a contig, features are auto-created and added to the consensus, and we can look at those in our Seq Builder. And then another way to bring in features is to blast regions and collect features from blast hits and add those to the consensus. And so, again, a number of nice ways to bring annotations into your project. And so we'll jump out of the PowerPoint here. So that's kind of an overview of what I'd like to do today. And we're going to start with a small project. So I'm going to open SeqMan Pro. And the small project, this is actually the demo data set that is provided with all of the SeqMan demos. And it is 14 trace files, ABI files. And usually I drag and drop these in, and they go into this unassembled sequences window. And I can go and look at the trace file. So I just double-click there. And I can expand the trace file and take a look at it. And you can see the electropherogram peaks. There's some options here. I can zoom in and out. So I have some nice control there. I can also look at quality scores. And if I zoom out too far, then we lose room for them and we lose them. So if I go back to quality, it kind of expands. So I can look at all the peaks. So it's a nice viewer for peaks. And you see this vertical bar. This bar is actually trim points. And so right now there's no trimming applied to these sequences. And you can see the limit on the sequences all the way to 741. I can manually trim sequences. So one way that some folks have used SeqMan is just to manually trim. So I just grabbed the bar and dragged it. And you can see that it's a little muted in color. That means I added a trim point. And it will be updated then in the limits range. And I could also go down to the back end, and I could just kind of eyeball the quality scores and select a trim point. So of course, that's nice if you have a project where you're trying to get that last couple of base pairs to get something to align. But of course, you don't want to do that for a lot of sequences. So we won't do that. There are automated trimmers. And the reason that I'm looking at trimmers now is that if you have a project that's going to require editing, before you do any of the editing, you want to be very certain that you've done an assembly at the right stringency that limits the amount of manual editing that you might have to do. And so trimming and editing really go hand in hand. And so you want to be very familiar with the trimming tools that are available to you. And SeqMan Pro can assemble ABI files, and it has its own set of options for cleaning up the data prior to assembly. And we'll also look at SeqMan Engine, which is used for assembling next-gen data, and it has its own set of tools for cleaning the data up prior to assembly. And so you want to be very familiar with how to trim. Now, if I just assemble as is. So I didn't specify any trimming options. I get a report file that tells me how things went together. And you see that went together very quickly. And so here's the contig. And I can see the ABI files here. I'm just going to scroll through. And you can see these red areas of mismatch. Okay, there's quite a few of them, and they seem to be on the 5' ends or 3' ends of some of the reads. This would be a 5' end here. And you can see there's quite a bit of mismatch there. It's not affecting the consensus that's generated. So we have the aligned reads here, and this consensus is at the top. SeqMan has a great consensus caller. It's really, really robust. I think it's the best out in the market. And so it usually can get it right even with noisy data. But you don't want to have errors if you can prevent them. I'm also going to show a strategy view. And this is more of like a genome browser type view. It shows us shows us where the reads are aligning, and it gives us a depth of coverage histogram. So I can see, that where they all overlap each other, we've got 12x coverage or something. And out on the ends, we just have one sequence. And I can double click and go right to the point in the assembly. Now, if I want to look at the trace data, I can click the twisty triangles, and I can see trace data. If I hold Control-Alt down and click, I can do all of them at once. And now I can also zoom in so I can see the peaks a little bit better. And I can also, again, show quality scores. All right, so I can go in and look at this data quite well. Now, those errors, one of the questions that you'll have are, is the data clean enough to give me an accurate consensus sequence, or am I going to have some problems in the consensus? And so there's ways to navigate the assembly rather than just scrolling through like this. And there's a little search window it's easily missed down in the left corner here. And if I click right on unspecified search, I get a window that comes up that allows me to search through this project to look for potentially areas that are interesting. And so one might be conflict. And so I'm searching for a conflict now in the consensus between the aligned reads. And so I'm going to search for anything where the consensus caller isn't quite sure what to call at that location. And so I can see here is a lowercase t, and that means some of the reads have a strong T, and some there's just a gap. It's missing. And so it's not entirely confident. Now, I could decide that this is some kind of a sequencing error. So one thing I could do is edit one sequence and add a T there. Now, I don't highly recommend editing the actual chromatogram, at least in the project. It doesn't actually change the original ABI files, but it just changes what's reported in SeqMan. So that's one kind of a micro edit, is actually directly edit the ABI. And in some cases, you can look at the peak and figure out why there's some confusion at a certain peak. Another option is to actually edit the consensus itself. So we can see we have an ambiguous here, and I might say, well, even though I see C's and G's there, if I decide that that is a G, I might override and just type a G in. So I select it. But now I'm noticing, boy, there's a lot of errors here. So if I go to conflict, and I'm just clicking on these chevrons that move me from the next conflict, I'm like, wow, that's a lot of errors. So I would stop at this point and say, you know what, let's try to get a better assembly here. I'm going to collapse this down again so I can see things a little bit better. And I'm going to make my quality scores go away. It's kind of how I prefer. I can say, oh yeah, look at that assembly. It's just, there's something here. So I might blast this. I might highlight a sequence and say, well, what's in my data here that's creating a problem, and blast it. I know what the problem is, but I'll blast it, and it's going to hit vector sequence. So this is a case where these trace files had some vector sequence that was untrimmed. We also see this in Ngs Project. So not so much the Illumina genomic data, but where we see these kind of N contaminants are things like transcriptome data, where there may have been a cDNA library kit or something where some primers, linkers, adapters are untrimmed and on the ends of the sequences, right? And they show up just like this, and then having millions of reads that have linkers like this, causes a more inefficient assembly and lower quality and all that sort of thing. So you want to trim back, identify problems, and trim them, and then go through all the steps of editing. So I'm going to just close this. And I'm going to hit delete now. And what happens is that Contig dissolves. And here's my sequences again. And I know that this has something called a Janus vector. And the Janus vector, you can see there's a little question mark. That means we haven't scanned for it yet. And so I can go under options, and I can scan all the sequences. So what I did there is I just did a trimming step. I said, look for Janus, and if it's there, trim it. And the check mark means that that vector is found in every read in this assembly. And now, again, there are ways to use SeqMan just as a trimmer. So you might have an instance where you really don't even want to assemble. You just want to clean up the data and send the sequences somewhere. And so I can do a contig, export sequences into multiple files or a single file, and send out the trim sequences. So that's a workflow that occasionally comes up. And now there's some options here. Under trim ends, I have different stringencies, and if I hover over them, what this stringency is a quality threshold. It's looking in a window along the sequence and trimming due to according to the quality. And I can manually define window sizes, all that sort of thing. I'm going to leave it on. I want to have some differences just for the webinar. So I'm going to go with a vector trim, but just low stringency, and scan them later. And now when I hit assemble, it'll do that last kind of a little bit of a change in the quality trim, and it's doing the vector trim. So now when I look at my contig, I'm looking at these ends, and I don't see as many. We'll scroll through. So we just reduce the amount of mismatch by a lot. And so now I might be ready. I might say, well, go back and do a high stringency assembly, see if we can remove some of these ends. But we'll go with this just for the webinar's sake. Okay, so I'm going to expand these out again. And I'm going to go back to my search. Now I'm going to look for a conflict in the consensus. And there I find a conflict. And so the conflict, it looks like there is a C in one and some slippage possibly in another. And I'm kind of looking at that peak, so it's a little bit of slippage. So I think that the consensus is right. And what's really nice about SeqMan is that the consensus caller factors in the quality scores. So it's going to look at the quality and say, we've got a high quality C here and nothing here, but we're still going to call it C, but we'll make it lowercase just so in case you may want to look at that. Okay, and then I can go to the next conflict. Another thing I can do is you see that these ends of these sequences have triangles on them. What's really nice about SeqMan, and before I worked at DNASTAR, I did a lot of manual editing like this with a program that when you trimmed, it was lost for good. And it really was a hindrance because I couldn't go back and extend trim ends. Now, when I extend this end, look what happens. All this mismatch, that's probably vector. In some cases, though, it's just low quality sequence. See what happens here. See there, now we're getting a bunch of ends there. But sometimes even a crummy sequence like that, you can get just a little bit more data to confirm your consensus. And that one doesn't work there. Let's look for another one. And you can see that's the only conflict in the consensus. So let's change our search terms and look for any differences. So it's a little. So this is where SeqMan makes a call based on quality, but other methods, like the majority method, shows ambiguity. And so here we can kind of look at some cases where there's some ambiguity, but SeqMan is making this call. And so I can kind of get faith in, is SeqMan making the right call here? So I can look at peaks. We have a little valley there with a T. So I'm just kind of looking at those and say, well, it looks like there's another one that's a T. Let's extend one, see what we have there. Another one of those valleys. But I think it's probably a C, so I'm good with that. But I might decide to make a change there. And I can, again, go ahead to the next one. And we have, again, a valley with a C call. All right, some ends there. So it's a nice way to move through the assembly. And so here's maybe I can extend here. Nope, I start getting more mismatch. So I keep running into that vector. So still haven't found a good spot where I can make an extension. Again, and again, if there's any changes that you need to make, if I decide that, well, I really think that's a G, I can go in and highlight, and I can make that change in the consensus. So again, this is the kind of micro-editing kind of capability, again, both the trace files and the consensus that are really nice, especially when you're working with ABI files. Now, once I'm done making the edits that I want to make, I can send this consensus different places. So one thing I can do is save the consensus out to a single file. And this dialog has changed, so for those of you that have used SeqMan before, make note of some of the options that are here. So I can save as a number of different sequence files, and I can save either the entire consensus or a range of consensus, and then I can also do things like include gaps, or if I have a reference sequence present, I can check this option. So this is really an important piece that gets overlooked fairly frequently, and that's if I had a reference sequence that I align to, by default, SeqMan will be forced to use that reference as the consensus, and it has something to do with the snip calling. So if I want to export something that is not the same as my reference, I check this box and hit save, and it'll save out. It'll recompute a consensus without the reference bias, so it's an important little checkbox to know about. Okay, so that is some of the micro edits. Now let's on an ABI file. Now let's go and look at what an NGS project might look like. So if I've got a bigger project with Illumina data, for instance, I'm not going to assemble that in SeqMan Pro. I need a more powerful alignment program, SeqMan Engine, which is designed to handle NGS data. And again, there are webinars on specifically this workflow, so I'm going to kind of move through this part quickly just to get to the project. But so if I'm going to do a de novo assembly or a genome assembly, I can pick a de novo option, or if I have a template, we can pick a templated option, and I'll show you an example of a templated as well. So Engine is set up so that you can make different choices in your workflows and name the project. This is going to be an E. coli K12 strain using MySeq Illumina data. And you'll notice the output options here. And it's important, depending on your project, to select the right output option. And so if you have a project that has under 10 million, you might be able to stretch this to 15, but if it's microbial in size, you can create this editable project to work with. If you have a project that is larger and it's de novo, then the BAM file, even a de novo BAM file, is going to be the option that you're going to want to choose. ACE files are we don't run into those too often. It's also an editable format, but it's not very efficient with next-gen data. So I'm going to pick a SeqMan Pro format that's editable, pick my read files, right, the technology, and then I can load unpaired or paired data. I loaded 2 FASTQ files here with a very tiny little actually there's not even an insert on many of the reads. They're nice long MySeq reads. And so here's the screen I really want to get to, and that's when I do a de novo assembly in particular, I want to make sure if I'm going to be doing a lot of editing and I want to build a consensus for a new strain that's as accurate as possible, I want to make sure that I trim those reads as accurately as possible before they get assembled. And so SeqMan Engine provides a whole host of different trimming tools. And for instance, if there's that adapter or vector that's contaminating my data, I can set and load in a vector file. And SeqMan Engine will do a vector adapter scan that is MER-based, identify those contaminating sequences and trim them off prior to assembly. And of course, there's also quality trimming, so you can, and these are quality score ranges set for Illumina data. If I loaded a different platform, these are going to change a little bit. The window sizes, we can do fixed end trimming. There's quality score independent trimming called trim to MER that is a good option if the quality scores aren't as accurate as in some cases the quality scores aren't as accurate as they need to be. So there's a lot of different trimming options here to really clean that data up before you start doing any manual kind of editing. Okay, so then we say okay, and I'm just going to breeze through this, and it's ready to assemble. And what we get is a .sqd file. It's going to be a lot bigger than the ABI file that we're just looking at. So I'm just going to quit this and open up the project. I think this is it here. So here's the de novo assembly. When you open these de novo assemblies, by default what you'll have is a report file that summarizes the assembly. It tells me how long it took, tells me which version of SeqMan engine I used, how many contigs I have, how many contigs to reach the genome length, the contig N50, average coverage. So I get all the metrics here. So and also when I make edits, the edits will be added to the end of this report file. And so so that's what we'll look at a bit today. So so we can sort the contigs different ways. So this is a big editable project. So I can sort by length, and I see we have some nice, really big contigs, and I can double click on them, and we can look at, and scroll through contigs. Right? There's no trace data here. There's still quality scores, but we don't have traces. All right, so go back to, and I can look at these in a strategy view as well. And so the strategy view now has got a lot more than our first project. We can see these peaks and valleys. The pair consistency is usually it's green when we have distances. With this data set, there's very little pair distance, so it's called inconsistent. They actually overlap each other, but that's not really a concern. It's just a nature of this particular data set. And so I can go through and look at coverage maps. And so when I do a de novo assembly, I'm really concerned, is this an accurate assembly? Do I have areas in these contigs that look, like maybe they weren't assembled properly? And so I don't want to have to look through all the contigs, but I might if I have 100 contigs and I'm trying to put this genome together, I may spend the time and look at each contig and then do some macro editing or some end editing. So for example, on this contig, I get these two sequences extending off the end, and there's a little bit of mismatch there. Not too big of a problem, but if there was more mismatch, especially at the join, where it joins this thicker area of coverage, I might decide, well, that might be a chimeric read that's contaminating, which does occur. So I might say, let's just highlight those areas and hit delete and trim them. So one thing I'll do is I might manually go through some of these contigs and do this kind of micro editing. It's not necessarily one sequence at a time, but I might highlight a group of them, and I call them the toenails, and kind of trim those off like that. Now what I just trimmed, I probably wouldn't have done that because I don't see mismatch. Well, here's one that is, okay, here's one where it's a thinner area. You can see we have, so these are kind of areas that sometimes are, you see this kind of thin extension. Sometimes that is actually a chimeric read, and I'd like to trim those. Okay, so that's another way to kind of edit contigs. Now, there might be errors in the middle of contigs. Now, with Illumina data, MySeq, it's really good data, so there aren't going to be a lot of errors in the assembly. An engine is very stringent. But if you look, you're going to find a couple places in each assembly where you might not want a contig in one piece. You might want to break it apart. And so I'm going to I know there's one in here, and I have to sort by name. So I'm going to sort by name, and let's just keep it in order here. So we're going to go to contig 257. And I'm going to go to the strategy view. And there's an area that would be concerning. And you can see that in the coverage map, we have an area where there's this red line is one read that is joining two areas with coverage. And it looks like there's quite a bit of coverage here, or excuse me, there's quite a bit of match, but it's small, and I might not trust that. I might say, well, let's break that apart and maybe try to confirm it in a way. Now, one way to confirm is to manually go in, just like we did the Sanger data, and extend ends. And I can see, well, that actually matches pretty good. I'm going to go to and there's some low quality, what happened is there's some low quality scores there that cause some of these reads to be trimmed back. And so it may just be, so I can extend. Now, if I extended and I saw a lot of mismatch, like that one has more mismatch, so I'm not so sure. I might look at a region like this and say, well, maybe that's, And we'll just go to the other side. And here we see a little bit more mismatch in there. So we can see why it trimmed back. So something's going on there. So I might decide to break this contig apart. So now is where we move to, rather than micro edits, macro edit. And I'm going to split this contig. And now you'll notice it's taken a little long. This is a pretty strong computer. It took a little longer there than it did with the ABI files. And the reason for that is there's many, many more sequences in this contig than there were with the ABI. So it's something to be aware of, is that when you start editing large files with lots of sequences, you might not want to do that on a laptop. You might want a little sturdier desktop so it can do those functions more robustly. And I'm going to go back to the project report, and I can see what happened. We split contig 257 at position 2862, and we created a new contig 276. And so we have two contigs now where we had one. So I just edited that contig. And now what I can demonstrate, so those two contigs we think may go together, but there's different ways we can confirm this. And here's a step in editing that is key when you have projects like this with a lot of contigs in them. When we go to the contig menu, there are things where I can align and force join and do things to the project. You want to resist clicking align contigs unless you're sure you want to align certain contigs together. This data has already been aligned to SeqMan Engine. SeqMan Pro has its own aligning algorithms. You don't want to have to realign all these contigs together with SeqMan Pro. SeqMan Engine has already done that. So what I want to do is more focused kind of alignment. So if I want to join these contigs together, what I want to make sure to do is lock this project down. And so what I'm going to do here is, I think I can just go up here, is lock everything first. So now when you lock contigs, you get this lock symbol. That means that these contigs cannot be realigned without unlocking them first. And so I'm going to go now and so, for example, if I wanted to figure out if those two contigs really should have gone together, I'm going to focus on them, and I'm going to unlock 257 and 276. And there's more than one way to do this, but I could use some of the tools here now under the contig menu. And you can see I can use something called extend contig ends. And extend contig ends is an algorithm that's doing what I had just done manually. I was manually extending contig ends by grabbing the little black triangle and extending off the end. But there's an algorithm that can also do that in mass. And so I can extend the ends, and you can see they're highlighted in yellow. I'm just going to go to the other one so you can see. And there's another extended end. So it's doing some of that kind of automatically. And so I can extend those ends. And now I can say, well, do these align together? And so I can go back now. And now at this point, I might be doing some gap closure. I might have decided, well, that was thin, that area. Maybe I did a PCR amplicon. I added another sequence to the project. And again, all the gap closure steps are, there's a whole webinar dedicated just to gap closure. So in an area like this, I might actually have added another sequence. I'm just demonstrating now I can break contigs apart. I can put contigs back together. So I'm going to go back to contig. And I can align a couple different ways. If I align contigs, it's only going to look at those two, and it's safe to align them. The reason that it would be unsafe to align all of them together is it is possible that that area is an IS element that occurs 50 to 60 times in this E. coli genome, and that there may be other contigs that share homology with those ends. So if I unlocked everything and said reassemble, I may get some reassortment that I don't want. So I just want to try to put these two back together so I can align these contigs. And so in my report now, it says it's merging them, the percent match. So again, a contig editing, I can put them together. Now another way to do this is I'm going to go back and split. Strategy view. So let's go back here. I'm going to split my contig one more time. Okay, so there's 257 and 277 now. Another thing to do is to make another order above contig level, that scaffold level. So I could say, let's make a new scaffold, and I can also maybe I think there's another sequence that I could add to a scaffold. So I can create scaffolds here and add more sequences to a scaffold. And in a scaffold now, there's multiple ways to do this, and again, this is covered in some of our de novo genome work, but if you have mate pair data, we can auto-create scaffolds. We have an automated genome workflow that will automatically create scaffolds and put all these contigs in a putative order, or I can do it manually like I've just done. The scaffold has another order of information called position. The position, and I can add a position, 1, and I'm just going to change that to, so however I get this information, I might think that they are in position 1, 2, 3, and I can sort the position column. And I can align, now there's another way to try to merge the contigs. Now I know that I added contig 264, that probably isn't going to overlap, but I can say, let's see if there's overlap now in these scaffolds. So there's another algorithm that's similar to align contigs, but it's align contigs and scaffold end to end. It is looking for overlap between adjoining contigs in this structure. So again, it's another way to merge contigs safely. And you can see that the one that I just kind of randomly picked, it didn't align it, but it did align our two that I had split into one contig. Again, I can edit a scaffold by moving, I clicked, and I can drag and drop that contig. Say, well, that one didn't fit in that scaffold. Maybe there's another sequence that I can add in. So contig editing, then we can build scaffolds. We can edit scaffolds as well, and so forth. So that's a lot of the different editing tools. Now, in the last couple of minutes here, I want to show you some feature handling tools. And so I'm going to go to a different project now. And there's not as much content here. So, and let's see, that's not that project. Let's see, this one. Okay. And so this is, again, the same data set, but aligned to a genomic template. And this is a template that I modified a bit, and we're looking in the strategy view, and you can see now instead of a bunch of contigs, we have one contig. It's the full genome length. And I'm looking in the strategy view, and I can see all these little hash marks, and if I hover, I can see that this is a gene feature. So we have upper and lower strand feature annotations now associated with the first, part of this genome. And so there's a couple different ways I can create features. So one way is to highlight a region. So I might just kind of go and select a region and go to the feature menu and create a new consensus feature. We'll call it Matt. And I can give it a description. I can define what the feature is, and we'll pick gene. Okay, so I've just manually created a feature. And there's a test feature I did earlier. So right now there's only 2 features on my consensus. It is largely unannotated, and this is a table then of consensus features. Now I can also so I'm going to close this. And I'm just going to close this window again here. And go back to my strategy view. Okay, so here's all the reference features. Let's see here. I want to get them to come back here for me. And I'm not getting them to pop back for me here. I'll move to the next annotation. Okay, so I can also bring in features by selecting a range and blasting it. And when I get the blast hits back, I can collect the features. So this takes several minutes to run, and I can blast ranges up to tens of thousands of KB, somewhere in that range. And I can look at the blast hits. I want to make that a little bit bigger here. Let's see here. We'll go to font size, make that more readable. And so I can look at my different blast hits, and if there's features contained within these blast hits, I can collect those features. And so what it's doing now is it's saying in the database blast region on this sequence that I selected, all these features are contained there. And so I can add those directly to my consensus feature. And so it's a way to blast and annotate. Now, another way that annotations are brought in is through the coverage report. So I'll go back here. Okay, and so in the coverage report, there are different regions. So this report lists areas that are deeper or thinner than expected, and they might be interesting. And you know, depending on what your analysis is, you may want to know where there's insufficient coverage or excessive coverage. And so I can go to project parameters, and I can control what's considered maximum coverage or minimum coverage. So I can set that parameter and save the file. And when I export the consensus to our SeqBuilder program, these locations will be annotated as feature types. Okay, so that's manual addition, blast, coverage features, and then there's the transfer over. I'm going to try that again. I might have gotten my, let's see here. I think I can. Get it back here. Excuse me. Oh, I think I know what's going on here. Is this project has an unmarked reference right now. All right, so I just marked my reference. And so I'm going to show the feature table. What I want to show is the feature table of all the reference features. It might be building here just a little bit. Come on, feature table. Okay. Okay, so these are the features from my reference now. So I had to remark it. I had forgotten that I had unmarked it. So here's all the reference features. Now I can go to the reference features and let's see here. Select a gene, and I have a couple different options. I can either take all the features here and move them to my consensus, or I can do just subtypes. With bacteria, of course, CDS overlap genes almost in a perfect correlation, so I may not want to transfer all of them. So I can select all of type gene and say, okay, take all the gene features now and transfer them over. And so I can go back and copy these selected now over to the consensus sequence. So now my consensus contains a mix of different features from a mix of different sources. There's those from the reference sequence, manually created features, blast collected features, and then what we don't actually see in this table are the coverage features. Now there's multiple different things you can do. I can take this going to have to select my contig here. I can save the consensus out into a single annotated file. Again, I have a reference in this assembly, so I want to make sure to do an unforced consensus or unmark the reference like I had done before accidentally. And so I can save this annotated.seq file out or a GenBank file. Another thing I can do is just send the consensus out to our SeqBuilder program. And so SeqBuilder then is our sequence editing program. It allows us to make circular maps, what we have visible here is a linear map. There's different feature types that are in this file now, and I'm just going to show you. Again, we started with an unannotated consensus, and now we have different features that we can apply. So, for example, here are gene features, and I'm zoomed way out, so I could zoom in, and there's Matt. Of course, SeqBuilder has the ability to move features around and do things like detach labels. All right, I can set default colors and stylings for all the features. Again, there's separate videos that cover what SeqBuilder can do, but we can see all these features at the front half of the reference. These are from the reference. Here's the one that I manually created, and I believe that these are the blast features here that are off in this little group here that I collected from blast hits. And then, of course, we have the coverage features. I'm going to make the gene features go away just so they pop a little bit better. And so coverage that exceed my 250x threshold, and there I can see some coverage features. And there's little blips here that I can zoom in on those. We have some little ranges there where we had some peaks, probably over IS elements, or well, we could probably turn this back on and maybe a ribosomal gene, I think. I'm not sure, but yeah, looks like a ribosomal gene where we had some peaks of coverage, so we can kind of interpret what some of those peaks in coverage are. So that's what all the material I intended to cover today. I know I went through a lot of stuff kind of fast. I'm sure there'll be some questions, so I will stick around here for the next few minutes and answer any questions that have come up. If I don't get to your question today, or during the webinar, I'll certainly answer you through email. So again, thanks for your attendance, and I'll take any questions.